So, biologics. Why do I think it's important to speak to talk about biologic? Because since the first ones, the first monoclonal antibodies, so the OKT3 it has been used for transplantation. There's been tons of biologics that are been developed, and if you look at the market value of those biologics now, is just skyrocketing. We can look at this graph. So Herceptin is a very good antibody. It's now very old, from '98. The omalizumab, 2003. So now we're almost 20 years that we have this antibody with us. And now there's every year more antibodies are produced. You have also the biosimilars that starts to be, and this one, the Umlicoclo, has been approved by the EMA, so it's the biosimilar for the omalizumab. And so we're going to be exposed more and more to these antibodies, obviously to reaction to them. Of course, when we start to talk about monoclonal antibody, we should disclaim some conflict of interest. I don't think I have important conflict of interest for this presentation, but I have to mention them. So if we start with a very simple thing, the definition. So what is a biologics? And I think there's two things you have to retain. The first one is, in contrast to chemicals, it's produced by a living organism. So I think that's very unique to biologics. And the second thing is, this is not small molecules like the aspirin, 21 atoms. These are actually very complex molecules. They can go up to 25,000 atoms. And so obviously the reaction to this structure of biologics is going to be very different from what we know as compared to normal drug hypersensitivity. The second thing, which I think is really cool for us as also a researcher, a scientist, a clinical scientist, is that this antibody can be identified quite easily from human. So it means that in my lab, for instance, we can take a birch allergic patient and we can try to sort the B cells, which are the memory B cells. We're gonna immortalize these cells with ABV, keep them in culture for a few weeks, and by ELISA we are now able to screen for the few clones who are making antibodies that are specific, for instance, to birch, and we can characterize them, and we can try to think of new therapeutical approach. This is, by the way, something that is currently done by Regeneron. You may be aware of that, but there are now clinical trials testing the hypothesis that whether you can neutralize antigens or allergen from people suffering from rhinitis or asthma. And so that means we have the opportunity to produce a lot, or to discover, identify, and characterize a lot of antibodies. And because these pipelines now are quite well established, it allows us to think of a lot of different new targets. And this is why I think in the future we're going to be even more and more exposed to biologics. When we talk about antibodies, we have to think about what's the structure of them. And it's not just about thinking of the target, but also taking into consideration a certain number of things. The first is the class. So you have to think about which subclass this antibody is belonging to. And the subclass is defined by the heavy chain, the constant region of the heavy chains. And obviously, if it's a IgG1 and IgG2, 3 or 4, it's not going to be have the same effector function. Another very important thing that you have to take into consideration is the glycosylation. These antibodies, depending from where they are produced, they have different type of glycosylation. And I will talk a little bit later on. The most famous problematic glycosylation for the allergists are the alpha-gal. And obviously, what you have to know is that glycosylation is going to affect the effector function of the antibodies, but also the half-life. Other than that, you also have to think that there is excipients. So we also have to discuss and think about which excipients are used to solubilize these antibodies. And antibodies are getting more and more complex. It means we are not just using a single antibodies. Sometimes now we're thinking of engagers, so B-specific antibodies. You can have synthetic proteins, be made of fusion proteins. You have P-specific antibody, trait-specific antibodies. So it's going to become more and more complex. And this is why it's very important to go back and understand which is exactly the structure of the biologic that we are prescribing. Okay, and then we have to think about the effector function. When we talk about effector function, we have to think of the target. And it's not just knowing the target, it's also understanding how it binds. Does it neutralize or is it not neutralizing? And what is the activity? Is gonna activate the receptor, gonna inhibit the receptor, gonna be a neutral activity? And I think these are a question we should address when we think about an hypersensitivity reaction. And then there is all the effector function coming from the FC dependent part. And so what you have to understand is that antibody can bind complement, and of course you can have a cytotoxicity that is dependent on the complement, but they're also gonna bind to FC receptors. And these FC receptors, they can lead to phagocytosis, and they can also lead to cytotoxicity through the activation of NK cells, for instance. And to make the thing very more complex, the FC receptor, there is a variety of them with different affinity, and every cell has a different combination of FC receptor, which can be activating and inhibitory. So it makes actually the full picture of a reaction to antibody could be becoming quite complex. Glycosylation, I mentioned it just before. I think glycosylation, it's one of the most common and most important post-translation modification of proteins. So this is something very important to consider. A very nice example is that if you take an antibody that is untouched, or you try to deglycosylate it, you can see the FC binding capacity of this antibody is almost completely lost, except for one of the receptor. And so there's also a lot of reactions that are related to this glycosylation. I want to here take the opportunity to remind you that we have proteins, lectins, in our body that can interact directly with this sugar. And if you remember the different pathway from the complement, so the complement can be triggered or activated through antibodies, but it can also be typically triggered through this protein, these lectins, and lectin can bind to carbohydrate and induce the complement activation and some hypersensitivity reaction. We are all, I guess you are all aware of the cetuximab story, the alpha-gal, how it comes that we have some antibodies that, so alpha-gal is a sugar, first of all, and this sugar is can be present and depends actually from the cell line that produce these antibodies. So if you have, and that was the case of the cetuximab, they use a mouse myeloma cell line, and this cell line actually has this galactosyl transferase in it, and naturally will make this antibody with alpha-gal at the end. And this is why we have now some people who are sensitized to the alpha-gal through tick bone, for instance, that start to gonna react at the first time when they are exposed to this antibody. So this is why it's also very important to understand which is the source of the cell line that is making these biologics. Another thing that is obviously important that we have to take into consideration is the excipients. Why do you have to take into consideration excipients? Because antibodies are often amphiphilic. That means they can aggregate. If you leave them alone, it means that if you have a protein that or an antibody that start to aggregate, it will start to denature, and you may have a loss of function. And this is why you need a surfactant. So you need to add into that some surfactant to stabilize this preparation. And every protein is different because the nature of the protein are different, and you don't need the same quantity of polysorbate to stabilize these antibodies. So just as a reminder, but polysorbate, they have so this hydrophobic chain, and they have this sorbitan that is surrounded from this ethylene glycol motif. And basically what the immune system gonna see first are really that, and they can make actually antibodies against the ethylene glycol. This is also quite known that when you have these excipients and you have probably antibodies, you can have complement activation, and this is something we can measure, for instance, with some drugs such as paclitaxel, for instance, where you have a lot of this polysorbate. So it's also depending on the quantity of polysorbate that you have in the drug to know whether you can trigger strongly or not the complement. you have also to take into consideration that every antibodies is actually not, they can have a different immunogenicity. So if you infuse now an antibody that is coming from a mouse, you will very quickly make antibodies against these antibodies, and the treatment effect will be lost. And so what people have started to do is to take, okay, what if we just keep the viable part or portion of the antibody, and we fuse that synthetically with a human antibody. And those are the mAbs that are called, we have with the Xi, such as rituximab, meaning that you have a constant part that is human, but the viable part of it is from a mouse, for instance. And this is obviously still immunogenic, meaning that you can generate antibodies quite easily against this viable part, because we are not used to see the sequences, these antibody sequences. And so therefore, people have started now to say, okay, we're going to take the whole framework of a human antibody, and we'll show you how we do that later on the next slide. And we just plug in the CDR region. And this is the humanized antibodies, which have this suffix "zu". And finally, what we're going to see more and more are antibodies that have been discovered directly from humans. And this is more and more easy, and FDA, Swissmedic are more keen to approve these antibodies because they have been already in circulation into patients, and so you have a safety profile that is more guaranteed. So if you want to graph, and this is something I've personally done, so this is why I showed this slide, but you can take the Herceptin antibody, which is a very stable antibody, and you can graft into it the CD region of another antibody from a mouse, for instance, and you can have this fusion protein, this humanized version of the antibody, and now you still get the specificity of it. It's not working every time, but sometimes it works, so you can keep the specificity, and you lose the immunogenicity because you have a fully human backbone on it. So when you look at an antibody and a reaction, I think this is the first thing which really have to be done, is to look what is the source of the production of this antibody. Is it a humanized, a human chimeric antibodies? What's the isotype of these antibodies? Is it IgG4? Is it an IgG1? It's not going to be having the same effect. Of course, you have to think of the target, and it's not just the target. You go a little bit beyond the target and how it works exactly, and taking into consideration not only what excipient is in there, but also the quantity of it. And with that, I think you can try to predict potentially cross-reactivities. So now if I go to the deep of the subject, hypersensitivity reaction, I have to acknowledge first review the most cited ones, I can say, in the literature from 2005, from Werner Pichler, and I think it's a nice one. So that's how you categorize the different reaction or potential reaction to antibodies in alpha, beta, gamma, delta, epsilon reaction. I think this is something still very useful today. You have to take into consideration those who are mediated by cytokines. We will talk about that later. You have those who are linked to a kind of hypersensitivity reaction. That means we have an immunization process behind that. It can be IgE, can be IgG. Nobody talks about IgM, but probably that IgM could also affect these type of reactions. And then there is all this gamma reaction, means that if you inject rituximab, obviously you can expect that with the loss of B cells, this patient can start to become immunosuppressed, and so you're gonna have some imbalance that's gonna be provoked by these antibodies. But with the advent of all the checkpoint inhibitors, we have also seen a lot of different type of reaction, autoimmune reaction, and we also start to see some allergic atopic disorders. I think we had, 2 weeks ago, one of the first cases where a patient was under Dupixent, and a few weeks later started to be on antibiotics and started a very severe dress reaction. So I think these are things that we can provoke by this immune cytokine imbalance syndromes. Then we talk about this cross-reactivity, the delta reaction. This is kind of off-target activity. I don't think this is very simple to always think of which are the exact cross-reactivities of the antibodies, very specific of the drug that you inject. And you have the last bucket of whatever reaction that is not immunological. Yeah, that's someone else who adapted from this. It's I think for those who watch the presentation can also read it. It's also quite interesting, just summarizing what I just said. And now you have this ADR-AC position paper who propose a slightly different way of seeing things. And so I'd like here also to take the opportunity to summarize what they propose. And so the classification here, it's really about, and we're going maybe a step back at, do we have immediate reaction and do we have delayed reaction? And from the immediate reaction, they look at what could be infusion-related reaction. These are at first infusion reactions. It's not very clear what the mechanism are behind. Is it a nocebo reaction? It's something that is self-limiting in nature. You can start again the perfusion. And then you have, when these things are a little bit more severe, you have more cytokines, and now we're talking about cytokine release reaction. I don't know why we should not talk rather on cytokine release syndrome, but it's obviously some overlap. More severe reaction, and then you have all these antibody-mediated reaction, can be IgE or now non-IgE mediated, and obviously as often you have mixed reaction. And then you have this more delayed reaction, serum sickness reaction-like, local reaction. I think there are quite a lot that can be also T cells, and sometimes you have more a severe T cell reaction or type 4 reaction that are also acknowledged. Do we have some biomarkers that can be useful? We always measure the tryptase, but maybe when we think about cytokine release syndrome, we should also look at IL-6, for instance. So there is a few studies suggesting that could be a useful measure to look at it, just to give also a sense that it's not only mediated by the mastocyte, but also by the cytokines produced by other cell type. We have skin tests, but we have to acknowledge that the concentration of the skin tests are typically very poorly defined in general. The general rule is to say that you can use it as a 1-to-1 for a skin prick test, and then you can dilute 100 times for the intradermal reaction. But many of these antibodies, because they have quite a lot of surfactant, they are viscous, very difficult to handle, and so the validation of those tests are just poor. But this is something we can also do, and here behind that, the idea is really to detect whether we have some type of immunization. All the reaction, I think this is also a useful marker, is to look at anti-drug antibodies. And there's different way to try to look at that. The most efficient way is to look at the IgG. Doesn't mean these are those who are responsible for the reaction, but it's a proof of concept that you made some specific immunization or reaction against the biologics. There's probably also some basophil activation tests where you could measure eventually activation. I don't know how far or how good this is established, but possibly we could also use basophils as a surrogate for this, the presence of this ADA. I hope I'm not too fast. In the ADR-AC position paper, so they really try to make the distinction between ADA-mediated and non-ADA mediated reaction, and so really looking at whether you have made or you have an immunization, an active immunization, we can explain the presentation of the reaction. From those who are not mediated by antibodies, of course they explain or talk about the cytokine release reactions, but they're also mentioning the CARPA, also the reaction that are mediated by the complement. I'm not quite sure whether it's purely complement mediated, but maybe you also need the presence of antibodies for that. But it also could be that the release of C5a, for instance, that activates secondary the mast cells, and you have also some degranulation through this mechanism. how do we should manage this type of reaction? I think it's very important so to have the characterization, the history, the type of antibody that has been used. We should obviously also consider the excipients, as I said. So far, we have mostly tried to look at the presence of ADA, these anti-drug antibodies. We have to try to see whether, if we can, make some skin tests to see if we have some clues about some immunization that is behind these reactions. And then the general rules is to say that if it's a severe reaction and that you have a proof of concept that you have a proof that there is a active immunization behind these biologics, you should think of an alternative. And really, if you don't have an alternative and then you have a poor prognostic associated to it, thinking of a desensitization process. And when you have no clue of an immunization behind these biologics and the reaction is not too severe, I think it's reasonable to provoke under medical surveillance. So if I have to conclude, and I'm happy to take after that all the questions, when we look or think about hypersensitivity to monoclonal antibodies, we have to think of the history of the prescription and drug allergy. I think this is super important. Something that is always forgotten and nobody really mention it is to really understand more deeply the characteristic of the biologics that are used. So the target, the subclass, the type, the glycosylation. Unfortunately, we don't have much tools to analyze the type of glycosylation, but this is probably something very important too. The excipients that are present, obviously. And then you have to define the type of reaction, whether it's local or systemic, whether it's immediate or rather delayed. You have to define the severity. And maybe look for biomarkers, not just the tryptase, but it has been mentioned that the complement activation, so the C5b9, there's some papers suggesting that the C5a could be also an interesting biomarker, but that's so far just an hypothesis based on not much evidence. IL-6 also could be a tool to distinguish between cytokine release reaction and hypersensitivity. Looking at ADA, I think we should work more on tools we can develop to identify them. So ELISA, it's a way, but there's other ways to detect them, maybe through basophils. The skin test, I think, is still useful. We can do them for non-severe reaction. We have to think of activation tests or LTT for a more severe reaction. And try with that to evaluate now the risk of cross-reactivity with the alternative monoclonal antibodies. And this is designed when we don't have a good alternative. Okay, with that I'm finished, I'm done, and I'm happy to take questions.
Thank you, Yannick, for this nice overview of the immunogenicity of biologics. Questions?
Patients do you see with these problems? Are they sent to you to the polyclinic or to the outpatient clinic?
They are sent mostly to the outpatient clinic. That's true. Yeah, these are patients who receive their antibodies, in the outpatient clinic, and they make a reaction in outpatient clinic from other specialty, and then they are referred to us because for the next one they want to know what to be done. But it's true that we don't have that many problems with biologic for hospitalization purposes. But we do, but I mean, it's.
You mentioned the IgG subclasses.
Yeah.
Can you tell me briefly advantages, disadvantages, IgG1 and IgG4, because these are the most often used subclasses?
Yeah, so I took a few ones for the type 2 inflammation, just to see the overview. The test pair is an IgG2. We have IgG4. So when you target a soluble factor, typically you want to neutralize, going for an IgG4 which is not going to bind the complement and somehow less poorly the Fc receptor. This is the rationale that is behind. But then between so those who are going to have the strongest complement activity are the IgG3 and IgG1, and then IgG2 is there's still some complement activation associated to it, but much less. And obviously there is also some Well, there is some patent issues, so they, to make sure that they have the product protected. But to estimate, what's the risk of cross-reactivity? If it's an IgG2 and then the alternative antibodies is IgG1 or IgG4, this is something you could take into consideration to say, okay, there's here the risk associated to cross-reactivity may be slightly lower.
Another question. So the development of ADA actually in clinic is quite frequent for TNF inhibitor, for example. And in most of the cases related to decrease of the efficiency of the therapy.
Yeah.
But you don't see many symptoms, if at all, but you see clearly because of this development an involvement of T cells and of B cells as well.
I think it's like IgE. You can have IgE, no allergy. You can be sensitized, and this is the same thing that occurred. The difference here is that if you have neutralizing antibodies, the ADA, because this is also what really do you neutralize now with your antibody the activity of the infliximab, and if not, then it's true that you may consider them as non-relevant. But it's a nice proof of concept that you have an active immunization. And if you have IgG, it could be that you have IgE2, and it could be that you also probably have some T cells are specific, because immune reaction never comes alone, or not frequently.
One last question.
Which lab in Switzerland can measure IgG or IgE? Because I have always the difficulty to find out who can measure ADRAC or
the CHUV,
I don't know.
So in the CHUV they make some for those who are validated for the infliximab. There's a list of ADA that we can measure, but are always not defined of which type of and there's not more behind this analysis. But this is something that the CHUV has started to do quite a long time. They were probably among the first. And now we have more and more people interested in that type of techniques, and I think these are very useful tools to, you can send, you have to know for which antibody. We are not looking at that for all antibodies. Those antibodies I just mentioned, we don't have the possibility. So omalizumab, dupilumab, there's no kits, nobody has validated that. It would take quite an amount of work, but it looks like that since the efficacy is not lost, or not that much lost in this patient, that we don't make that frequently ADA for those specific antibodies. Yeah.
Yannick, in patients with chronic urticaria who are under omalizumab, do you see frequently a sensitization to polysorbate 80 or not?
So I've seen a lot of sensitization with the vaccine, but not with the. I've not checked, so I don't know.
Last question, Werner.
I wanted to give him a difficult question. Yes, finally you waited for it. Yes. I mean, the problem we get sometimes. The question if there is an immune reaction to a biological.
Yeah.
And I find some, but I don't know if it means something.
Yeah.
Because we have many of us make IgG or something like that, which is completely harmless, not damaging, and of course we cannot differentiate that in our assays. Do you have a clue out of it? So that's a difficult question.
No, I think the assay should really be thought about the neutralization. So we should think of functional assay. I think that would be very useful, not just measuring. Now we know that very well for the COVID and the vaccine. People just look, do you have antibodies? No, it's not just about having antibodies. It's what's the quality of them and what's the binding. Yeah. Yeah. Yeah, that would mean we should think of cell lines, or that you can use with different type of Fc receptors, and we made the antibodies. We made some IgE. We tried to test them, and we think that the glycosylation also affect the binding to the Fc receptor. So there are a lot of things. It's so much more complex than we think.